In the spring of 1985, in a quiet laboratory in Germany, a young scientist named Svante Pääbo opened a small glass vial and did something no human being had ever done before. Inside that vial was a fragment of tissue taken from the mummified body of a child who had died in Egypt more than 2,000 years ago. Pääbo believed, against the near-unanimous skepticism of his colleagues, that somewhere inside those brittle, sun-baked cells, a molecule had survived. Not just any molecule. DNA, the very code of life, waiting in the dark for 40 centuries, sealed inside linen wrappings and desert heat. When his results came back, they seemed to confirm the impossible. He had recovered ancient human DNA. It was the doorway he thought he had opened was an illusion. And for the next 4 decades, that failure would cast a long shadow over one of the most tantalizing questions in all of science. Whose blood truly ran through the veins of the people who built the pyramids? And did any trace of them still run through ours? It is a question that sounds simple on the surface, but it cuts to the heart of identity, empire, migration, and myth. For centuries, the ancient Egyptians have been claimed, reimagined, and argued over by nearly every culture that came after them. Their statues have been sketched by European scholars who quietly gave them European faces. Their history has been used to fuel political arguments about race, ancestry, and origin, often centuries after the people in question had already turned to dust. Everyone wanted an answer. Almost no one had the tools to find one. Because unlike the cool, stable permafrost of Siberia or the icy caves of Europe, where ancient DNA has survived in remarkable condition, Egypt is one of the most hostile environments imaginable for genetic preservation. The heat that mummified bodies also destroyed the very thing scientists needed most. High temperatures accelerate the breakdown of DNA at a molecular level, shredding it into fragments too short and too degraded to read reliably. And it gets worse. The embalming process itself, involving chemicals like sodium carbonate to dry out the body, may have made things even harder for DNA to survive, not easier. For decades, the working assumption in the scientific community was almost a kind of surrender. Ancient Egyptian genomes were considered a lost cause, a mystery permanently sealed by sand and time. If you wanted answers about who ancient Egyptians were, you turned to hieroglyphs, to pottery, to the bones themselves, and what they could tell you about diet and disease. But the deepest layer of information, the genetic blueprint hidden inside every cell, remained out of reach. Then, in 2017, a team led by geneticist Johannes Krauss at the Max Planck Institute decided to try again, this time armed with tools Pääbo could never have imagined in 1985. Next-generation sequencing technology had matured to the point where scientists could read millions of short DNA fragments simultaneously and then digitally reconstruct them, like reassembling a shredded document one microscopic scrap at a time. Crucially, this new approach could also detect a telltale signature of true ancient DNA, a specific pattern of chemical damage that accumulates over centuries and simply cannot be faked or accidentally introduced by a careless lab technician. If the damage pattern was there, the DNA was almost certainly ancient. If it wasn't, you were looking at contamination, exactly the kind of mistake that had derailed Pääbo decades earlier. The team turned their attention to a site called Abusier El Melek, an ancient settlement and necropolis along the Nile in Middle Egypt. There, they collected samples from 90 mummified individuals, spanning an extraordinary stretch of 1,300 years, from the height of the New Kingdom through the Roman period. What they were about to uncover would rewrite the opening chapter of a genetic mystery that had remained closed for 40 years. Despite the odds stacked against them, the team successfully recovered 90 complete mitochondrial genomes. The genetic material passed down exclusively through the maternal line, and remarkably, they also managed to extract genome-wide nuclear data from 3 individuals, an achievement that had been considered nearly impossible for material this degraded. It was the first reliable, high-resolution look at the genetic makeup of ordinary ancient Egyptians, not pharaohs, not royalty, but the everyday people who lived, worked, and died along the Nile. And what the data revealed cut straight, through centuries of assumption and speculation. The ancient Egyptians in this sample closely resembled ancient and modern populations from the Near East, especially the Levant, the region spanning modern-day Israel, Lebanon, Jordan, and Syria. It was a finding that immediately reframed how to think about the ancient Nile Valley, not as an isolated genetic island, but as a crossroads deeply entangled with the populations of Western Asia. But the most startling twist came from what didn't change. Egypt, over the course of that 1,300-year window, was anything but politically stable. It endured invasion after invasion. Libyans pushed in from the west. Assyrians and Persians conquered from the east. Greeks arrived with Alexander the Great and stayed for centuries under the Ptolemaic dynasty. Then came the Romans, absorbing Egypt into an empire that stretched across 3 continents. On paper, this reads like a recipe for genetic upheaval, wave after wave of new rulers, new soldiers, new settlers pouring into the population. And yet, the genetic data told a different story. Despite this relentless political turnover, the core genetic ancestry of the mummies remained remarkably stable across the entire period studied. The pharaohs changed. The language of administration changed. The gods on the coins changed. But at the level of the genome, the population beneath it all held its shape with startling consistency. It suggested something almost counterintuitive about how conquest actually works. That armies and administrators can rewrite a nation's politics far more easily and quickly than they can rewrite its underlying population, and that the genetic identity of ordinary people can prove more durable than the empires that rise and fall around them. Then came the twist that would ignite public fascination and no small amount of controversy. When the researchers compared their ancient samples to the DNA of Egyptians living today, they found something unexpected. Modern Egyptians carry noticeably more sub-Saharan African ancestry than the ancient mummies did. In the ancient nuclear genomes, sub-Saharan ancestry was almost undetectable. But in modern Egyptian populations, somewhere between 15 and 20% of mitochondrial lineages trace back to sub-Saharan Africa, a substantial and measurable shift. Something happened after the Roman period that had not happened before it, something that reshaped the genetic makeup of the Nile Valley more profoundly than 1,000 years of foreign invasion ever had. What could account for a shift this significant? The researchers pointed to the centuries following the fall of Roman control, a period marked by increased trans-Saharan trade routes, improved mobility along the Nile corridor, and more darkly, the expansion of trans-Saharan slave trade networks that moved people, against their will, from sub-Saharan Africa into North Africa and the wider Islamic world over the following centuries. It's a sobering reminder that the genetic story written into modern populations is not just a tale of ancient migration and conquest. Sometimes it is a record of trauma, coercion, and forced displacement, quietly encoded into the DNA of people who may have no idea that history left this particular fingerprint behind. The scientists were careful to note their sample size was small and their conclusions preliminary, but the pattern was clear enough to demand attention, and clear enough to overturn a simplified, static idea of Egyptian identity that many people, for many different reasons, had long assumed to be fixed and unchanging. For all its brilliance, though, the 2017 study came with an asterisk that nagged at researchers for years afterward. The mummies at Abusier El Melek, remarkable as they were, only reached as far back as around 787 BCE, deep into what's called the Third Intermediate Period, long after the age of pyramid builders, long after Tutankhamun, long after the Old Kingdom that most people picture when they imagine ancient Egypt. The genetic data from that earlier, formative era, the age when Egypt itself was being invented as a unified civilization, remained a total blank. Scientists were staring at a genetic record with the first, most crucial chapters torn out. That would remain the case for 8 more years. Then, in the summer of 2025, an announcement from the journal Nature would finally fill in some of that missing history, and in doing so, deliver the oldest and most complete ancient Egyptian genome ever recovered. The story of how it happened begins not in a laboratory, but over a century earlier in 1902, in a remote village called Nuweirat, roughly 265 kilometers south of Cairo. There, archaeologists working on Douar, a British excavation team, uncovered an unusual burial. Instead of the ornate wooden coffins associated with wealthier tombs, this individual had been sealed inside a large ceramic pot, itself placed deep within a rock, cut tomb carved into a hillside. The remains were eventually shipped to England, passing through the Liverpool Institute of Archaeology, surviving even Nazi bombing raids during the Second World War, before finally settling into the collection of what is now World Museum Liverpool. For well over a century, this man rested quietly in storage, his identity, his life, and his origins completely unknown, just another set of old bones catalogued among thousands of others. What nobody realized, for more than 100 years, was that this particular burial had accidentally created something close to a perfect genetic time capsule. The individual died sometime between roughly 2855 and 2570 BCE, an extraordinary window in history that straddles the very end of the early dynastic period and the dawn of the Old Kingdom, precisely the era when Egypt's earliest pyramids were beginning to rise from the desert floor and the kingdoms of Upper and Lower Egypt had only recently been fused into a single unified state. This burial predated the era of artificial mummification becoming widespread practice, and that, paradoxically, may have been the secret to its survival. The sealed ceramic vessel, combined with the stable microclimate of the rock-cut tomb, appears to have protected his remains from the punishing extremes of heat and humidity that had destroyed so much genetic material. Elsewhere, as one of the study's co-authors, archaeologist Linus Girdland-Flink, would later explain, the combination of the pot burial and the surrounding rock-cut tomb created a stable environment, precisely the kind of conditions ancient DNA researchers dream about but almost never find in Egypt. In 2021, researchers extracted samples from 2 of the man's teeth, one of the last. Strongholds where fragile ancient DNA sometimes survives, protected inside dense enamel long after softer tissues have disintegrated. Leading the effort were Adeline Moores Jacobs, working at the time as a doctoral researcher at Liverpool John Moores University, and Pontus Skoglund, a paleogeneticist at the Francis Crick Institute in London, who had founded one of the first high-throughput ancient DNA laboratories in the United Kingdom. By Skoglund's own admission, the team's expectations going in were low. Egyptian remains had disappointed geneticists again and again. Mummified individuals, he would later say at a press briefing, are probably not a great way to preserve DNA at all. But this time, something different happened. 2 of the tooth samples contained enough authentic, undamaged ancient genetic material to do something that had never been achieved before for anyone from this period of Egyptian history: reconstruct an entire genome. Not a partial one, not a handful of scattered genetic markers. A complete whole genome sequence achieved at just over 2-fold coverage, meaning the DNA had been read enough times over to give researchers real statistical confidence in what they were seeing. It was, without exaggeration, the oldest Egyptian DNA ever successfully sequenced, older than the Abusier El Melek mummies by more than 1,500 years, reaching back into an era that scientists had assumed was permanently lost to the ravages of time and climate. The very first thing the genome revealed was elegantly simple. A Y chromosome analysis confirmed the individual was male, but it was what came next that stunned the research team and would eventually reshape our understanding of how ancient Egypt came into being in the first place. When researchers compared his genome to reference populations from across Africa, the Near East, and Europe, they found that the majority of his ancestry closely resembled that of early farmers from the Neolithic period of North. But his genome carried a second, unmistakable signal layered on top of that North African foundation. A meaningful proportion of his ancestry, the study estimated somewhere in the range of 1/5 of his genetic makeup, traced back to populations from the Fertile Crescent, the historic cradle of civilization spanning modern-day Iraq, Iran, Syria, and Jordan, the very same region where Mesopotamian civilizations were independently inventing writing, agriculture, and urban life. For decades, archaeologists had suspected a connection like this existed. They had long observed shared cultural fingerprints between early Egypt and Mesopotamia, similar pottery styles, comparable early writing conventions. Well, the gradual spread of the potter's wheel, an invention historians believe originated in Mesopotamia before making its way into Egyptian workshops. Trade routes carrying domesticated plants and animals back and forth across the Fertile Crescent and into the Nile Valley. All of it pointed toward contact, toward exchange, toward 2 of history's earliest great civilizations quietly influencing. This genome changed that. For the first time, scientists were holding direct genetic evidence, written into the chromosomes of an actual person who lived through this transformative era, confirming that the cultural links long suspected between the Nile and Mesopotamia were not simply the product of merchants trading goods at a distance. Real ancestry, real family lines, had crossed that distance too. And then the story took a deeply human turn, one that had nothing to do with statistics or population genetics and everything to do with a single, ordinary life, lived thousands of years. Civilization defined in the popular imagination by golden pharaohs and monumental man was somewhere between 44 and 64 years old at the time of his death, an unusually advanced age for someone living in that era, when life expectancy was often cut brutally short by disease, injury, or childbirth complications. Standing at roughly 160 centimeters tall, a little over 5 feet, his skeleton bore the unmistakable signs of a lifetime of grueling physical labor. Severe arthritis had settled into his joints. His muscle attachment points showed the kind of pronounced development seen in people who perform repetitive, strenuous work for years on end, and his posture and bone structure hinted strongly that he had spent countless hours sitting, reaching, and twisting in ways consistent with one very specific ancient profession: a potter. Yet here lies the twist that made the entire discovery even more remarkable. Despite this evidence of hard manual labor, the nature of his burial suggested he held a position of genuine social status. He wasn't discarded into an anonymous mass grave. He was sealed with deliberate care inside a ceramic vessel, placed thoughtfully within a rock-cut tomb, a burial practice reserved for people the community considered worth remembering. It painted a picture entirely at odds with the rigid, simplified hierarchy that pop culture has attached to ancient Egypt, one where laborers toiled anonymously while god-kings and their elite courtiers received all the honor and permanence. Here was a working man, his body worn down by decades at the potter's wheel, who nonetheless received a burial signaling respect, even reverence. Using this combination of skeletal, archaeological, and genetic evidence, forensic anthropologist Caroline, and this is very likely only the opening chapter. The scientists behind the New Wayrat discovery were candid about the limitations of working with a single, extraordinary genome. One individual, however remarkable his preservation, cannot speak for an entire era or an entire population. As Morez Jacobs herself put it, piecing together the clues from this individual's DNA, bones, and teeth allowed the team to build a comprehensive picture of one life. But future samples from ancient Egypt will be needed to determine, including a facility at the National Museum of Egyptian Civilization in Cairo, led by molecular geneticist Yahia Gad. Researchers are hopeful that the next major breakthrough will not take another 40 years to arrive, unlike the 4-decade gap that once separated Pabo's flawed first attempt from the field's eventual redemption. Because that is, in the end, the real story hidden inside every one of these discoveries. Not simply who the ancient Egyptians were, but how astonishingly difficult, how nearly impossible it was to ask their DNA the question at all. For 40 years, an entire civilization's genetic history sat locked behind a barrier of heat, chemistry, and time that scientists believed might never be breached. It took a failed experiment, a wrongly celebrated breakthrough, decades of technological patience, and finally, an improbably well-sealed pot in a hillside tomb to begin cracking that barrier open. What we've learned so far is already reshaping how historians and geneticists talk about ancient Egypt, replacing old assumptions about a fixed, unchanging people with a far richer, more dynamic reality, one written not in stone or hieroglyphs, but in the molecules passed down against all odds, from a potter who lived and worked and grew old on the banks of the Nile nearly 5,000 years ago. His face has now been rebuilt from the bones of his skull. His ancestry has been traced across 1,000 miles of ancient trade. Roots. And somewhere out there, in tombs not yet opened and sands not yet disturbed, more of his neighbors are almost certainly still waiting, holding fragments of a story that is nowhere near finished being told.